So again, what is the genetic significance of meiosis? So after a cycle of meiosis and fertilization, the chromosome number is maintained in sexually reproducing organisms. So you maintain the concept of 2n to the next set of progeny, 2n. Because of the number of possible chromosome combinations, there are many gene differences between paternally derived and maternally derived chromosomes. The way they line up, you can have variations during metaphase as to how they line up. And that's what we're going to look at here in just a moment. Therefore, the nuclei produced by meiosis will be genetically quite different from the paternal cell and from one another. What that means is that if we look at two pairs of molecules chromosomes, and so this is one dyad, here's the other one, and so this creates the tetrad. They line up, they have in a synaptimal complex, they've lined up. Here's the other one down here. They can line up this way during metaphase 1, and so they could be this way, or they can pair with this yellow is over here, this way. So this could be all mom's DNA, this could be all dad's, but it could line up this way, where this one goes to that cell and this one comes to this cell. So this is just looking at fruit fly, and if we do this, we have two of these. You take it to the number of 4, since there's 4 sets of chromosomes, we have 16 different combinations how they can line up here. Now let's look at the human. 2n to the 23rd, 2 to the 23rd. We have, for every time you produce in a gamete, we have over 8 million possible different combinations. So yeah, think about that. That allows us to have the variety of different individuals that are being produced. Now you will see in some situations where children look very much like their father or look sometimes like their granddad or something like that. Well, this is the case here, where you have the combination of these when they come together. So 8 million different possibilities for a sperm, 8 million different possibilities for an egg. That's a lot of variation that's out there. Now, throw on top of that crossing over. DNA exchange that occurs, and it occurs in all of it. All of your products are crossing over. In that one, you have crossing over between maternal and paternal chromatids that in you that's occurring right now to produce the gametes for your children. During meiosis 1 generates still more variation, and so that final combination, there's no way to calculate it. It's astronomical for each one of those. So, when we're producing sperm, it's spermatogenesis, or producing eggs, it's oogenesis. So spermatogenesis, you go from a spermatogonium and you produce the spermatozoa. And so you go from 2n to 1n. Now in oogenesis, you're focusing on eliminating cytoplasm and get it down to a fusiform shape so that it can move. Here, you're focusing on getting rid of the extra DNA, maintaining the cytoplasm. So an egg is going to be fairly large versus a sperm. And it's needed for that to get through those initial stages of development. In humans, we got to get from the ampullary region of the oviduct, which is where fertilization occurs, and we have to get that actual zygote. It has to form into a specialized structure called a blastocyst, and it has to come down through here and then implant. So that means it has to have enough material already in that cytoplasm to get it down there to implant into the uterus. So its job is to get rid of the excessive DNA, which are called the polar bodies that are out there. So in sperm, they are long flagella with the DNA built into the head structure. They have a specialized structure that's designed from the endoplasm, the Golgi apparatus, that contains digestive enzymes to be able to penetrate into the egg, because the egg's going to have a lot of protein coat on the outside. So it's regulating how many sperm get to it very quickly. And so the sperm will penetrate through the protective layers. In human male, spermatogenesis is a continuous process. A mature human produces several hundred million sperm per day. So you can see these are going to be haploid cells. Oogenesis, basically, unlike spermatogenesis, is a division. Oogenesis is asymmetric, meaning that you're not producing even amount of cytoplasm and structure like that. Here you're trying to eliminate the excess DNA in the form of polar bodies. So they produce these polar bodies, they set it off here. There's the first polar body, goes through division. There's a second polar body, and some of these will divide twice. And then you end up with a haploid egg. That's oogenesis. Now, as I said before, in female animals, it occurs in the ovaries. Early in development, diploid oogonia produce diploid primary ovocytes. In humans, we produce about 1 million, a woman produces about 1 million primary ovocytes per ovary, are produced before birth. They will then crawl into the ovary from outside, and they will initiate meiosis 1, but they will enter into a dormant stage during that prophase 1 period. So they are arrested at that stage and will become sexually mature once every month when the individual are released from this, when the individual becomes sexually mature. At puberty, they will periodically be activated to progress through meiosis 1. Only one cell per meiosis becomes an egg. And the division of meiosis 1 is asymmetric, producing 2 haploid cells of unequal size. You'll have a large secondary oocyte and a small polar body. And the secondary oocyte will go through meiosis 2 and be released into the oviduct. And that's called ovulation. Now, what is fertilization event? So, the secondary oocyte is going to be fertilized in the ampullary region of the oviduct. When that happens, that meiosis 2 is then going to be completed. And a haploid egg and a secondary polar body are produced. The haploid egg and sperm nuclei then are going to fuse. This is a unique thing. You can actually see what happens here. This is showing that particular secondary oocyte sitting there waiting for a fertilization event, and you're going to see a membrane blow up right here. So here we are. They're going to move around until they find the right egg that's going to be kind of individual egg. And then what will happen, you'll see the sperm be released into it. And when the sperm get released into it, the sperm are going to come here, and you're going to have multiple sperm trying to penetrate that egg. There's a big protein coat on it, on the outside. And when it gets there, here we go. Watch over here, you're going to see a membrane form that's going to blow out. Here comes the sperm. You can see them here wiggling around here. See them all around through here? One of them is attached already to here, and this is actually going to be the one that's going to penetrate through the egg. And you're going to see there it is, it's starting to form. There's a balloon starting to form here. Right in there, it's going to be ballooning out, and it's going to spread all the way around and push all the other sperm off of the egg so they can't fertilize it. Just starting to form. Where is it? I thought I saw it, but I guess not. Should be right in here. Ah, there it is, it's starting to form right there. Notice how the sperm are being pushed outward right there. See, that one's being pushed out. Now these are starting to be pushed out. It's going all the way around. And it creates a structure called the zona pellucida. And the zona pellucida is going to be an area that's going to protect the egg, now called a zygote since it's been fertilized, and keep it from implanting in the oviduct. And it allows cell division to occur through here until you get a blastocyst. Now, during that initial stage of fertilization, that was kind of slow there. There's actually a fast process of blocking the other sperm, and we can see it that when the sperm actually fertilizes the egg, we actually have an explosion or release of zinc, and it's caused by the release of calcium. And this is sort of the fast block of this. So what happens here is you can see these flashes. They put in a compound that's going to respond to the zinc. We're not sure exactly why the zinc does this, but you can watch more of this later on. But what happens there is they're able to actually see that the bigger the flash, the better the egg is in its fertilization. And this can be very useful for in vitro fertilization to identify particular eggs that have been properly fertilized so that they can select those for implantation. And you can see that spark occurring there. So that shows in animal eggs how those particular ones, but we also have to talk about plants. Plant structures, they produce sporophytes, and an anther produces the male gametophyte. So it's going to be produced up here. That's going to divide to where it actually has a nucleus for the tube cell. The tube is going to be what's going to penetrate the ovary, but it also creates 2 nuclei over here that are going to be sperm. Those are going to eventually be able to get to what we call the egg. The egg is down here, and then you have a diploid structure to here, which is called the polar nuclei. So one of the actual sperm is going to bind to that to create the endosperm, which is going to be part of the seed for growth, and the other one's going to bind to the egg to produce the next individual. So these have 4 megaspores produced in this particular one. And they go through the gametophyte generation to produce the sperm and egg, and then they have the sporophyte generation, which is 2N.